And my name is Gunilla Karlsson Hedestam. It's my pleasure to introduce the first keynote speaker of this meeting, Dennis Burton. I think he's fairly well known to most of you. He's local, he's based here at Scripps. And just a brief introduction, Dennis did his PhD in Sweden, actually, having done his undergraduate training in Oxford. He then was working in Sheffield for a while before late Richard Lerner recruited him to Scripps. So I guess it's about 3 decades that you've been here at Scripps now. And the task at the time was to develop methods to screen and identify antibodies from libraries, most phage display libraries in those days. And this started a really productive long career of working with antibodies mainly against viruses. Dennis was the first to clone a broadly neutralizing antibody against HIV, and this was followed by the identification of many potent broadly neutralizing antibodies against HIV, and as well isolation of antibodies against several other viruses, most recently against SARS-CoV-2, which we'll hear about today. Dennis is also a major leader in terms of leading big consortia. He's led the Neutralizing Antibody Consortium for many years. He's also leading a very large grant, the CHAV-D grant at the moment, that will lead into clinical trials for very innovative vaccine approaches. He is on the steering committee for the Reagan Institute in Boston. He is a board member of the Antibody Society, and as I understand, was very supportive of the AIR community joining the Antibody Society, which has been very helpful for the community. And with that, I think I'll just let you speak, Dennis.
Yeah, thanks very much, Nilla. Sorry about the delay. It could be quite interesting here as I kind of contort around to try and use the pointer at that screen. But thanks to Jason and Pam and Jamie and Felix and the AIR board for the opportunity to speak. So, you know, our lab has worked for many years, as Nilla said, on broadly neutralizing antibodies, mostly to HIV, really with the intent to drive forward the design of an HIV vaccine. And we've continued to do that, and we are doing that, and we're getting quite a few molecules into the clinic. But also over the last 2 years, like all the rest of you, we've been faced with this pandemic. And what I thought I would talk about today was what we've been doing on broadly neutralizing antibodies to SARS-CoV-2 as a sort of starting point again, really, for how we might move towards pan-coronavirus vaccines, which I think seem to be gaining more and more interest and traction, judging by the various quotes, as more and more variants of concern arise and people kind of realize that maybe, you know, COVID's not yet over. So, to the virus then. So, as you know, we see this all the time, every night actually, on US TV. And we know that neutralizing antibodies target those spike structures, which has this configuration, a trimer. And what I'm basically going to be talking about is neutralizing antibodies to the receptor binding domain, shown in blue there, and the top of the molecule, that's in an up conformation, and then the bottom of the molecule, the S2 stem helix. So we first got samples from COVID patients. I think it was a very early April 2020. There were a few around before that, but we didn't get them. So we got some samples from some folks who were infected in San Diego, UCSD, and we did our usual thing, which was, you know, isolate large numbers of monoclonals with single B-cell technology using the S protein as and RBD as probes to pull out the antibodies. And we, you know, a good thing about that was if you were you know, you got special permission, then we could still get into this place down the road here at Scripps, because we were working on COVID. So actually, we could continue some of our HIV research, but it really was very intense effort. And so we had folks from Scripps, from AIRR-V, and from UCSD, all working on generating these antibodies and testing them and so on. And actually, I think we got panels of antibodies pretty much at least parallel with, and maybe before many others, because it took us, I think, less than 2 weeks when we got the PBMCs to start looking at some of the antibodies. It was actually absolutely phenomenal, because people were all working in parallel. And what we found very early was that there was some just great neutralizing antibodies in the PBM made by these donors. Some of the best antibodies we'd ever seen. So they were neutralizing with IC50s in the nanogram per mil, single-digit nanogram per mil range. I just boxed one there called CC12.1, which became something of a prototype. And it was really staggering how, and other labs, very at the same time got similar results. So when we looked at these antibodies, they were, most of the most potent ones were directed to the RBD, to the receptor binding domain. And working with Ian Wilson, as we always do, we could show that they had the very similar footprint as ACE2, the ACE2 receptor, and neutralization would be mediated by inhibition of virus binding and entry. What was, I think is still surprising in some sort of ways, but was noted very early on, was that that there was a lot of variation between SARS-2 and SARS-1, the original SARS, in this receptor binding domain that's targeted by these antibodies. So the variation is shown in orange here, and more conserved regions shown in a grey colour. So almost kind of intuitively the opposite of what you might have thought. There's a lot of variation in the ACE2 receptor site. And this meant that virtually all of the antibodies that everyone was isolating early on were specific for SARS-2 and did not neutralize SARS-1. And again, I think we were actually the first to do this, to show that the monoclonal antibodies isolated from donors were protective in an animal model. And without going into too much detail, we were really using the Syrian hamster at that time. And if you had enough antibody on board, the animals didn't lose weight and they had low to non-existent viral titers. So there was a dose dependence, and the antibodies were clearly protective. Now, one thing that Laura Walker and I had written on earlier, 2018, was that increasingly folks are isolating antibodies with really remarkable potency and breadth. Now, usually, and HIV is an example here, usually you have to work pretty hard to get antibodies of this superb quality. You have to look through many different donors, and then you have to mine deeply into their repertoire to find these, what we loosely term super antibodies. I didn't make that cover, the journal did. I don't particularly like it, but there you go. But I think it's worth saying that these super antibodies fell straight out of SARS-2 donors, for lots of different people, which was really unusual. And that, I think, is a testament to the fact that these antibodies, it turns out, are actually quite close to germline. And humans are, what this does here is to just show you the different sites that are available, or a grouping of sites by Chris Barnes in Pamela Bjorkman's lab. And you can see they've classified the RBD into these 4 classes. This is the more conserved region, which will be more interesting for broadly neutralizing antibodies. And this is the receptor binding region, class 1 and class 2. And the class 1 antibodies, like CC12.1, our antibody, are typically, or higher, the highest representation is in the VH353 germline. So, and that, as I said, requires very little somatic hypermutation to neutralize quite effectively. So actually humans, we're very well set up to deal with SARS-CoV-2. When it first came, we were actually, you know, had repertoires that are very favorable. I'll show you something quite different in macaques in a moment, but you know, that's part of, you know, people said, well, this is a very easy virus and so on and so on. And at first, you know, it certainly appeared in that way. But, you know, having worked for years on HIV, we kind of have some sense that these viruses, you know, they don't just kind of roll over and let you make great antibodies and, you know, give up. They, you know, evolve. And here was, you know, a massive pandemic that's going to infect billions of people, tens of millions that are immunocompromised and are probably going to allow the virus to, you know, visit different solutions to escape immunity. So, and I'm kind of vaguely proud, I suppose I should say, that even in this early, May 2020, Laura and I wrote this piece for Cell Host and Microbe, where we sort of said, yeah, you know, it's working well now, but, you know, don't, in a way, underestimate this virus, that there is the possibility of mutation and so on and so on. And we should start to think about vaccine design and neutralizing antibodies and vaccine design based on more rational strategies, based on looking at more broadly neutralizing antibodies. And we followed that up later with Eric Topol, who's also here at Scripps. And this is a more political kind of statement about how we need absolutely vast amounts of money to deal with the pandemic preparedness, which I think is true. And actually there was, I read an article by Rino Rappuoli, which I would recommend to anyone, from last month, in which he quoted a paper that estimates that the total cost by 2025 of the pandemic will be $28 trillion, which is, you know, I mean, I don't know. I mean, how do I know? But there are, you know, economists and folks that do those kind of calculations. That's sort of sums that people are saying. And, you know, so preparedness in the billions of dollars is really, it's a drop in the ocean, but we'll see. Okay, so this is our basic starting point, if you like, for rational vaccine design, or if you want to call it reverse vaccinology 2.0 is what Reno calls it, or other people call it structural vaccinology. But the basic concept is that you, even for the most difficult pathogens, you will find protective antibodies somewhere in some people. If you mine deeply enough and you look at enough people, you'll find antibodies that would be protective if you could induce them through vaccination. And so our argument has always been, well, antibodies just recognize molecular shapes. So let's make those antibodies, determine the molecular shapes, and use those molecular shapes in immunogenic form as vaccines. And that's what we're sort of been thinking about. And that's why we go after the broadly neutralizing antibodies, because they're going to tell us how to make pan-viral vaccines. So in terms of coronaviruses, I have 3 different topics to talk about, and I'll be relatively brief. on each one, but I think they all are quite interesting from different angles. And a lot of this work, and I'll emphasize this again, has been led by Rais Andrabi, who's really done a phenomenal job. Okay, so let's start with the RBD, broadly neutralizing antibodies, from recovered vaccinated donors. And I think this is now in press. And so what we noted, and others noted, quite a few different folks have noted now, is that recovered, and this is all, you know, a little while ago, so this is before we had 3 immunizations. This is 2 immunizations or infection and another immunization. So what we noted was that recovered vaccinated donors had better titers against SARS-2 than just infected or vaccinated donors. And if we looked, and this is our interest, of course, in terms of cross-reactivity for sarbecoviruses, so these are sarbecoviruses, sequence related, so SARS-CoV-2 and PANG-17, the pangolin virus, are 1B viruses, and then SARS-CoV-1 and WIV1 are 1A viruses. And if you look here, you can see, and there's a stark contrast between SARS-2 and SARS-1, so infected folks don't make much SARS-1, nor vaccinated 2 times. But if you're infected or recovered vaccinated, then a fair proportion, and these are actually positives, a number of folks make reasonable, measurable cross-neutralizing titers. And they also tend to be better against different variants of concern. So these seem to us to be a good source to start looking for RBD, broadly neutralizing antibodies. And that's what we did. We did our usual things, a single cell, and so on, so on, so on. And we isolated many monoclonals, some of which are shown here, just to show you that you can get, you know, very potent neutralization. I mean, it's really potent. You know, these are 120, 40, 40, 10, 10 nanogram per mil across the top there. So those are really broadly neutralizing, potent, broadly neutralizing antibodies. And they very potent against different, some of them are very potent against a full range of variants of concern, including Omicron. And Omicron really threw a spanner in the works of antibody efforts sort of worldwide. I mean, all the companies, or most of the companies' antibodies fell once Omicron emerged. And it is an outrageous virus in inserting all sorts of mutations. And it certainly killed all the VH353 antibodies. So the antibodies that we make, you know, naturally, that don't require very much mutation, that go for the receptor binding domain and are very potent, of course, they have exerted the selection pressure. So that's where, you know, the escape has occurred first. And, you know, the CC12.1 that we made, I'm struggling a little bit to see in kind of parallel here, but I think it's here. CC12.1 no longer neutralizes a whole series of variants of concern, or Omicron. And Omicron generally escapes a lot of antibodies and escapes quite a few of these, but not all of them. Some of them are still neutralized quite well by Omicron, by the antibodies. So if we work with Ian, and again, so this is, you have to remember, we're not like after individual monoclonal antibodies to use for therapy. What we're interested in is panels of antibodies that help us to define sites that we can then use for immunogen design to try and design vaccines that will offer protection, broader protection. So we had a whole series of antibodies here, and they defined a footprint on the RBD that's shown here, any one antibody doesn't cover all of that, but this is the footprint. And we figure that, you know, we'll try and incorporate that footprint into immunogen design. And that involves, for example, trying to negate responses that would be the VH353 to the receptor binding site. So perhaps, for example, putting in extra glycans and or resurfacing the molecule between different immunogens used in a sequential strategy. So I'm not going to go there today, but this gives us the kind of clues that we need to have. And then just to round it off, we always look to see if the antibodies do what they're supposed to do. And this is work with Ralph Barrick, and this shows you, and Lisa Grolinski, and this shows you that single antibodies protect against SARS-CoV-1, SARS-CoV-2, and another sarbecovirus, SHC014, that's the most distant of the other sarbeco viruses. So, you know, the single antibodies can protect against multiple different sarbecoviruses, which, you know, gives you enthusiasm to pursue designs based around these antibodies. So I think I've probably already said all of this. The one thing I didn't mention was that there are certain V and D gene motifs that we see enriched in these RBD antibodies, broadly neutralizing. There's a D region containing YYDXXG, I think. That is a common motif. And once you get those kinds of common motif, you know, that's what Bill Sheaf has been using a lot in the HIV context to try and design immunogens that will pull out a motif like that. So that's all valuable information in terms of trying to design immunogens that might elicit RBD, broadly neutralizing antibodies. Okay, so onward to macaques, which was really quite interesting and a bit surprising to us. So we immunized very early on macaques with just S protein, just crude S protein with the 2P mutations for stabilization. And we used a very strong adjuvant from Darrell Irvine called SMNP. It's sort of similar to Iscomatrix and similar to, it includes QS21, so similar to ASO1B. So very strong adjuvant. And to our surprise, what we got in the sera of the macaques with just 2 immunizations was very strong SARS-1, SARS-2 cross-neutralization. And that was quite distinct from COVID serum that, you know, we'd seen at that time, single infections where you got little reactivity with SARS-1 or RNA vaccinee sera from 2 vaccine shots. I think you see that more coming up, the more shots you get. And then we looked at mouse sera, and they showed a bit more breadth, but not as much as the macaques, which was really quite interesting. And so, you know, we cloned those antibodies, and we got antibodies that were quite cross-neutralizing. You're going to see a couple of tables like this. I'm not going to belabor them, but you'll see pretty good antibodies with pretty strong, potent neutralization right across all the variants of concern there, including Omicron and, you know, across different sarbecoviruses. So some pretty impressive antibodies. And we looked at these antibodies, and there was a very strong enrichment for a macaque germline gene, VH373. So, you know, we ran that through the databases, and the closest human germline was 353. So, you know, we thought, great, that's it. You know, it's monkeys have been very clever. They've managed to recognize the same site as 353, but just in some way that, you know, these mutations don't have the same impact. Actually, there's a kind of lesson here, because that was completely wrong. Although the 353 is the closest, these antibodies actually recognize a totally different region to the 353 predominant receptor binding site antibodies. They recognize the side of the RBD in these are two different antibodies for Ian structures, and they come close to the RBD, but they're not the RBD. They're recognizing primarily a different, more conserved region. So I think that, you know, that's quite interesting for us, because again, it defines more conserved, another region that's more conserved, overlapping with the ones we have, but nevertheless interesting. But I think it also is somewhat, you know, there's something of a lesson there that you can't necessarily assume. Let's say I think in a way, if it had been me and if I'd have taken the immunogen and I'd immunized macaques and I'd have got good breadth, I would have said, yeah, you know, right on, my immunogen's really good. It's going to induce breadth. But, you know, macaques are sitting with a 373 germline gene that's kind of well equipped for that breadth. We're sat with the 353 that's good for the receptor binding site. So, you know, at least on the face of it, and there are all sorts of caveats to this. And, you know, we did use a very strong adjuvant. We don't have all the controls to compare macaques to humans. You know, obviously we don't have a lot of human data in exact comparison, but nevertheless, I think it's an interesting set of observations. So finally, we come to the S2 region down the bottom of the spike, and we've been working quite intensively on this for the last while. So we started out on this when we were looking for cross-reactivity for SARS-2 donors. We were looking at cross-reactive B cells between SARS-2 and the seasonal coronavirus HKU1. And you can see there are donors who have cross-reactive B cells, not with the alpha coronaviruses, only with the beta seasonal coronavirus. So we used those donors as a source to get at possibly cross-reactive antibodies, and we isolated panels of those. And we found that when we did that, that they were directed to the base of the spike, as shown by negative stain EM with Andrew Ward. And we took one of these antibodies that appeared to have cross-neutralization, and indeed it neutralized broadly, not very potently, but broadly. And I think this is the first S2 antibody in the literature, broadly neutralizing, and it neutralized broadly sarbecoviruses and also a whole set of variants of concern. And I think it also neutralizes now BA2, although I think there is some loss of potency there. So there is the target. We identified a peptide as being crucial for the binding. So we did lots of peptide scanning, found a peptide that was binding with high affinity to the antibodies. This antibody, sorry, CC40.8. Ian's lab solved the structure, and it had a helical structure, the peptide corresponding to the stem helix, pretty hydrophobic, lying just as shown here between the heavy and light chains. This is the helical structure. So that was nice. That was just one antibody, but that got us to then go and look for Oh, sorry. We also showed that this antibody was protective. We've now switched to the a mouse model, the human ACE2 mouse model. And what was notable here, I don't know whether this is going to show up so well, but what was notable here is we're still getting a level of protection even at a dose of only 10 micrograms to the mice. We normally use 300. So this was sort of indicating to us that this antibody is actually working in vivo better than we would have predicted from in vitro neutralization. So if you took CC12.1, for example, the VH353 antibody, at 10 micrograms, it would have been pretty weak. We normally wouldn't titrate it down that far. So, you know, there's some indication that this antibody what we say is it punches above its weight. And that's a phenomenon we've seen in HIV, that antibodies that come in very close to the membrane, and I don't know why, seem to work better in vivo than you'd predict from their NUC titers. So next thing, and again, this is totally illegible, but I'm just going to tell you, what we then did was to isolate lots of these different antibodies, stem helix antibodies, using selection based on the S protein from SARS and MERS. So we're now trying to go beyond sarbecoviruses or even seasonal viruses into MERS. And we got some antibodies, and you probably can't see it very well, but there are some antibodies in the far right-hand column there where you can see some relatively low numbers for MERS. So we've now got some antibodies that do cross-react between the sarbecoviruses and MERS. And as you'd expect, they neutralize very well different variants of concern, Omicron included, BA2 actually included, not sure about BA4 and 5. But, you know, that's kind of what you'd expect because the Omicron mutations, none of them are in this stem helix that we're aware of. So these are very interesting antibodies. So again, we've worked with Lisa and Ralph Barrick and in their mouse model. And basically what this, and again, I'm not going to bore you with all the details, but by several criteria, including weight loss, hemorrhage, respiratory function, the antibodies, 2 of these S2 antibodies protect very well against SARS-2, SARS-1, and MERS. So, you know, that's pretty broad. You know, so there are broadly neutralizing antibodies out there that will protect against these viruses. So, you know, it could be really quite valuable to try and generate vaccines that would act broadly against beta coronaviruses. I think you'd have more chance of picking up, let's say, some new emerging coronavirus. So these are the, and I've probably belabored this too much, but these are the conclusions from the S2 stem helix antibodies. I don't think I need to reread that. Let me just focus on the overall conclusions here. So it's pretty easy to generate broadly neutralizing antibodies actually to conserve regions of the RBD. It's not as easy as the receptor binding site, but it's not that difficult. But I must say, I think, you know, Omicron has to worry you that, you know, it's amazing what this virus can do with this receptor binding unit, the RBD. I mean, it can really put in a lot of different substitutions in there and still be pretty fit. So I think we'd be wise to have an alternate strategy to RBD, even with the broadly neutralizing antibodies recognizing RBD. And I think that's the S2 stem helix. What we don't know yet is in humans how good they would be at protection. Their NUC titers are not as high as the RBDs. They're more like 100 nanogram per mil, the best neutralization compared with, single-digit nanogram per mil for, like, say, the antibodies that were originally used in the clinic. And again, there are certain V and D genes that are enriched in these antibodies. So there are clues for immunogen design based on careful structural examination and protein design. So I think I just wanted to, really emphasize the contributions that Rais made, which were absolutely huge. He had a little baby boy last week, so he's really on top form at the moment. And then, so the acknowledgements, so there was Rais's lab, my lab, Tom Rogers did quite a few of the animal studies initially, Ian and Andrew for all the structural work, Delhi developed a newt assay, Brian's done all sorts of analyses of the antibodies. The SOC lab, IRV, is particularly crucial in the first efforts that I described. And then, Ralph Barrick's lab, we've been working closely with in showing these antibodies are effective. And I guess I didn't acknowledge the funding, which, is that we have this large CHARV grant and they allowed us to do COVID work with a supplement. The Gates Foundation was very generous, and Reagan Institute, and obviously we're all at Scripps. And that's it. Thank you. Sorry, I should have said any questions. Jordan. Yeah. No. I don't recall there being many insertions, and I think the average CDRH3 length's a bit longer than usual for the most potent ones, which would be one maybe negative factor. I don't remember a lot of insertions and deletions, not 100% sure about that. And the somatic hypermutation is fairly low, I think, relatively, not greatly different from the usual.
Hi, so thank you for this beautiful talk. I had a question concerning the fact that there are clearly enriched motifs, as you show, that may contribute towards this, these antibodies being broadly neutralizing. So do you think it would be possible to make or build a predictive model to assess the extent to which an antibody can be broadly neutralizing based on these motifs? Do you know whether there's been some work?
I think the one I mentioned, the YYD, I think there was a search through the literature, and quite obviously not all of them, but quite a few of the pre-existing antibodies that were known to be broad had that motif. So that was kind of support for the motif. Actually, Brian probably knows more about this than me. That's true, isn't it? The YYD motif was seen fairly broadly yeah. Ethnicity. So Nilla should be answering that.
Just responding to your comment that some of these neutralizing antibodies are germline. I don't know. How
well, I mean, I think we all have VH353 mostly. I don't know what the allelic penetration is in it, but Nilla will tell you.
353 is actually, you know, all the alleles of 353 have the motifs in the germline V, you know, the NY, SGGS. So all of the, and they're present in all populations that we've looked at around the world. So that's, yeah, it's kind of, you know, most people will be able to make those kinds of responses.
Right.
So if you would speculate a little bit, do you think it's going to be easier to make so going back to the rational vaccine stuff, right? So do you think it's going to be easier to make an RBD immunogen that masks or whatever that the immunodominant 353 thing and lets you focus on the broad site? Or do you think it's going to be easier to somehow get more and more potent S2 antibodies with some sort of S2 targeting immunogen?
I think the latter, but not, you know, by a huge amount. So I wouldn't give up on one. But I kind of, you know, that it's a linear sequence. I mean, I'm sure you can block it, you know, I mean, if you probably, you know, there are conformations or glycans that could cause, you know, quite a loss, but it's not under pressure now, I don't think, you know, because people don't make, you know, titers of S2 neutralizing antibodies are pretty low. So I don't think there's a lot of pressure out there. But with the RBD, you always feel that there, you know, there could be a fair amount. And some of these, you know, the more vaccine shots you get, I think the more, you know, and the more infections you get, the more that you can see these broad antibodies to the RBD coming up quite a bit more. I don't know. I mean, in some ways, I think it's an almost frightening, massive experiment ongoing, you know, with billions of people infected. I think I said this already, you know, tens of millions immunocompromised. I mean, it's just the perfect setting to, you know, find the escape solutions to all the, you know, all of the, almost any antibody you want to come up with. But maybe you'll pay a fitness price at some point. Then, you know, then it'll, but, you know, I think BA4 and BA5, I think they're even more infectious than the original Omicron variants. So I don't know where it stops.
First of all, congratulations on your instrumental work, which I hope prepares us for the next pandemic. And particularly, you've already had the insight to look at pan-vaccination strategies. I wonder if you could speak a little bit more to what sort of reception you got following this pandemic, and particularly from public bodies, the NIH, the FDA, on what exactly would they do to protect us from future pandemics, and in particular, what do you expect from industry in terms of novel tools? What are the tools that you need to enable you to take this further and to develop better vaccination strategies, which are far broader, in particular with some of the escape mechanisms you already can see now?
I mean, I think the single biggest factor is, you know, you've got to make a big investment in a lot of basic research. I mean, you know, to design these immunogens takes a lot of basic work. I'm not sure that industry is going to want to do that unless governments put in, because it seems like with COVID, you know, there have been huge amounts of, unbelievable amounts of government money's gone into, you know, the vaccines and the antibodies and the drugs and so on. And so you know, I might be being cynical, but I would think that there's a certain expectation from industry that, you know, if you're going to do it, then you expect that kind of investment from governments. I mean, I think there are initiatives ongoing now. I mean, I don't know them all, but there are initiatives out of the NIH to talk, and a lot of talking about viral vaccines and pandemic preparedness, but it needs translated into, and directed as well. What you don't want to see is just like huge amounts of money just going everywhere in some kind of scattered fashion, which, I think there's always a little bit of a tendency to do that, when faced with a crisis, but, I think there are certain strategies here that can work and a lot that can't.
Kind of a follow-up more from a science-based question. Given that we've got hundreds of millions of people now vaccinated with the major vaccines, to try and implement immunogen against a pan-coronavirus, do you think existing immunodominance is now going to be a problem?
Yeah, it could be. It could be. I mean, I think that's one of the sorts of things you have to, in your modeling, you have to take into account. We're actually doing that to a degree, sort of, if you're trying to design a pan-virus vaccine, then you immunize animals that are already have titers to, the original SARS-CoV-2, and see if it makes any difference. I think if you can probably override it if you, and also there's some evidence from emerging evidence from Facundo Batista that if you have too much of an antibody already existing, then you're going to block new antibodies, especially to that site. But if you have lower levels of antibody and not to that site, then that can actually enhance the response. So there's some kind of there's some kind of balance there, so maybe you do your pan-coronavirus vaccine when the original vaccine titers have gone down. It's conceivable that you would do it like that. Then you've got all sorts of complications because vaccines are always like, just vaccinate 1 million people and it'll just be the same in every one, and it won't. So I think there's a lot to learn though as well. I mean, I think that's another great thing is that people are really learning a lot from this pandemic.
I just thought we should not keep you too long, Dennis.
No, I'm
if there's
fine.
any more questions, we can take 1 or 2 more, and then I guess there's drinks outside. One over there. Okay.
Thank you. Very naive question. Why trying to find broad antibodies more than using a mix of very potent antibodies, but more specific?
I mean, that is an alternate approach. The trouble with that is you're gonna have to anticipate lots of different variants, and you might get something that works, with this variant, then there'll be another variant, and then there'll be another, and then there'll be another. And it can be very hard to actually cover them all. And for HIV, it's totally impossible because there are so many variants. You'd have to, For this, you might make better arguments that you could, and obviously now they're immunizing with Omicron, for example, Omicron mRNA. But my understanding so far is, at least now I might be wrong on this, but my understanding is that Omicron vaccination, I mean, it gives you a bit better titers against Omicron, but then they're not as good against some of the earlier variants, which could then, come back again, presumably. So, I think it's a more temporary solution, that just putting in multiple variants.
Okay, so we have the last question.
Hi, thank you for your talk. I was wondering, what were your thoughts about the lifespan of the neutralizing antibodies? Because both...
Where are you? I can't see.
I'm here, sorry.
Oh, okay.
I can stand up.
Thank you.
Sorry. Yeah, so both natural SARS-CoV-2 infection and the current vaccines seem to produce short-lived antibodies.
Yeah.
And this leads to frequent vaccination and boosters required, which is even more frequent than flu vaccinations. And of course, this involves costs and mistrust in the population. So I was wondering, what are your thoughts on the potential lifespan of a broadly neutralizing antibody and how the research field could improve on this?
Yeah, I mean, I think you're talking about the mRNA vaccines, which have, you know, for the neutralizing antibody titers, they're somewhat short-lived relatively, you know, they're certainly short-lived compared with, you know, live attenuated vaccines where, you know, the antibody titers can stay up for a very long time. I think there's a lot of work to be done on mRNA technology to try and extend the lifetime of the neutralizing antibodies. But even then, you know, that's still about, you know, the high titers of neutralizing antibody, probably what's required for symptomless infection, you know, so you might get a little bit, but probably practically nothing. But I think much lower titers and B-cell memory and T cells, they are still protecting you much further out, you know. I think people are still protected against hospitalization, at least against the original strains. I mean, it gets harder when you get to some of the variants, but, you know, I think the vaccines are still doing reasonably pretty well against, you know, hospitalization and death. So the question is, what do we want out of the vaccine? You know, are we content with not dying and going to hospital, or do we not want to get sick at all? I mean, the trouble with getting sick at all is then you transmit. And I think that probably is going to fire up a lot of work on mucosal vaccination, because that's what you've got to do. You've got to somehow stop replication in the upper respiratory tract, which is pretty hard. I don't think there are any vaccines that are very good against respiratory pathogens in preventing upper respiratory tract infection altogether. You know, I mean, I think, you know, many of the respiratory the vaccines will stop you getting, you know, stop you getting pneumonias or really sick, but stopping upper respiratory tract infection is very hard, because it's hard to get enough antibody up there. You know, when you've done a systemic vaccine, you know, the amount of antibody that's going to get up here is quite low, so you're still probably going to get symptoms. So I suspect one thing that's going to happen is more work on mRNA to try and extend half-life, the longevity of the induction of antibodies, and a lot more work on mucosal antibodies. One of the things that is perhaps a little bit concerning lately that I'm hearing more about is, you know, folks are getting infected and they're taking Paxlovid straight off the drug, and then they're getting infected again about 6 weeks later. And I don't know. I mean, I've heard like 3 or 4 of these kind of. It's kind of, you wonder what's going on there. You know, there's still, you know, there's so much still going on. It's, you know, I mean, I was in the UK like a month, 3 weeks ago, and you know, they're just completely over. You know, people just don't. It's just regarded as completely and utterly over. It's finished. That's it. There's no. But it kind of isn't.
All right, I think we should give Dennis another round. Thank you so much, Dennis.